Hey guys, welcome to Victory Education. This is your Narsiman, and the topic which we are going to talk about is the organelles of the cell. As you might be aware, the cell is the basic structural and functional unit of any organism on this world. So basically on this topic, we are going to talk about little detailed on the organelles of the cells such as mitochondria, plastids, cytoskeleton, ribosomes, and flagella cilia. So this is basically for an higher standard students to learn in detail about these organelles. So let's get into the video. Mitochondria is called as a powerhouse of the cell because they produce energy, which is nothing but ATP. So mitochondria can be seen on different shapes and structures, and some cells will have an abundant amount of mitochondria and some cells will have less number of mitochondria. The reason behind is the cell which needs more energy will have more mitochondria in them, and the cell which does not require much energy will have the lesser amount of mitochondria. So mitochondria are basically double layered. One is the outer membrane and another one is the inner membrane. The jelly part of the inner membrane is called as a matrix. So the mitochondria is made up of phospholipid bilayer. They generally call it as an outer phospholipid bilayer and inner phospholipid bilayer. Those are semi-permeable membranes and they just allow the small molecules such as sugar molecule. One of the important thing is the mitochondrial matrix is just like a thread. So they form a cristae formation when they just keep growing. So that's one of the key structure of the mitochondria that makes them as one of the biggest organelles of the cell. If the matrix is going to keep extending, then the cristae formation will be higher and higher, and the mitochondria size will keep enlarging. And the space in between the outer and inner membrane is called as an intermembrane space in mitochondria. So the next, as I told, is the mitochondria is the one which is going to produce the energy, which is nothing but ATP. So how they produce is the sugar molecules get into the matrix, and there they combine with oxygen to produce the energy. And apart from the ATP and energy, what are the other things you can see on mitochondria is the ribosomes. Ribosomes helps in protein synthesis, and you can find the granules. And one important thing you find on mitochondria is the DNA. Deoxyribonucleic acid. So if you're going to compare a mitochondria, you can say like mitochondria is just partially dependent on the cell. The reason behind is most of the things they can produce their own proteins, they can produce their own energy. So they don't depend more on the cell by itself. Mitochondria role on a cell is very important. Mitochondria almost plays entire cell life, even from the cell birth, the cell division and the cell death. Everywhere mitochondria plays a very key role. So the next important organelle we're going to talk about is the plastids. The plastids are generally divided into three: chloroplast, chromoplast, and leucoplast. Chloroplasts are generally green in color. They produce as chlorophyll, and that's the basic key for photosynthesis. The another key thing for chloroplast is they also stores and produce glucose. And second thing for chromoplast is any plant or leaf where you find a color, yellow color or orange color, it's basically because of the chromoplast. They are the pigment, and most of the times you can see them on the flower and fruits. The last but not least is a leucoplast, which are colorless, and they are basically used for starch storage. Now, let's see in detail about the chloroplast structure. Chloroplast structure you can easily compare with the mitochondrial structure because chloroplast is also a double layer, which is an outer layer and an inner layer, and you can see sugar molecules inside the inner layer. One of the key functionality you can see on the chloroplast is the outer layer of the chloroplast is a freely permeable membrane where almost all items or all molecules can be freely go in and come out of the outer membrane of the chloroplast, where the inner chloroplast is a semi-permeable membrane. The holes on the inner chloroplast are very small, so that smaller molecules can, like sugar, can go in and come out, but not all the molecules. Chloroplasts can be abundant in number. The chloroplasts can range from 30 to 200 in a higher plants, and you don't have any definite numbers for chloroplast. Chloroplasts can be in various shapes such as cup shape, grill shape, ribbon shape, and oval shape. All higher plants have oval shaped chloroplast. So the chloroplast structure in detail you can see here. One important thing you want to say on chloroplast is the thylakoid. Thylakoid is a key where this electron on the photosynthesis is going to be captured from the sunlight. And you can see the outer membrane, inner membrane, and intermembranous space, the same structure which you see on the mitochondria. Apart from that, what are the other key things you can find is the lumen and then granum and lamella. So let's talk about what these are in detail. Thylakoid lumen. Lumen is something which is inside the thylakoid. And if you're going to have a segment of thylakoid, like one after the another, if you're going to arrange them, that's called as a granum. Between two arrangements, the connectivity is called lamella. So the key thing you want to remember here is the thylakoid. Inside the thylakoid, we call it as a lumen or thylakoid lumen. And bunch of it, we call it them as a granum. And the connection between one thylakoid and the another, we call it as a lamella. This is again for your understanding what is the thylakoid system is. An arrangement of thylakoid is a granum, and between one granum and another granum, the connectivity is called stroma, lamella, or just lamella, and this entire connectivity is called as a thylakoid system. Basically, you can just imagine a plate as a thylakoid. Then if you're going to just keep one plate another after another, and for example, you have 4 or 5 plates together, then you call it as a granum, right? So that's the easy way to remember. Next comes the ribosomes. Ribosomes are very unique and small organelles of the cells, but they are very important. In fact, endoplasmic reticulum gets its name as rough and smooth with the ribosomes. If you have the ribosomes attached to the endoplasmic reticulum, then they named as a rough endoplasmic reticulum. If the ribosomes are not attached to the endoplasmic reticulum, then you call them as a smooth endoplasmic reticulum. A ribosome structure is really very small and very easy to remember. You can just remember a zip. The upper part is called as a large subunit and the lower part is called as a small subunit. How the ribosome structure works is like the amino acids gets inside the ribosomes and you have mRNA getting inside the ribosomes. They connect each other and they forms the proteins. So as you might be aware, the amino acids are the key base elements for the protein synthesis, and you have either mRNA or a DNA which is getting combined with the amino acids to produce the protein synthesis. That's why we call ribosomes. The basic definition of the ribosomes are they convert the genetic material into usable proteins. Generally, RNA and DNA are the genetic materials. They convert them to the form of usable proteins for the cell. Next is a cytoskeleton. Cytoskeleton are basically for the rigidity of the cell, and they are basically formed of 3 important parts: microfilaments, microtubules, and intermediate filaments. Okay, so basically if you're going to imagine a house, okay, how you are going to build a house? If you're going to build a house, you want to have the construction a pillar, right? So inside the pillar, what you will have? Iron rods. So those iron rods are the cytoskeletons. So without these cytoskeletons, the cell will not have its shape and size. So basically, cytoskeleton is for their rigidity. Next is the cilia. Cilia is just a small hairy structures which you can find on the micro level organisms, and they are basically used for locomotion. Next is the flagella. Flagella is also mostly seen on microorganisms and they are also used for the locomotion. Let's see how they locomote. The flagella will have a direct locomotion. However, the cilia will have a twisted locomotion. So now we're going to see an inner side, like a cross section of the cilia and flagella under a microscope. So on your right hand side, you can see a diagram which explains about the cross section of a cilia or a flagella. Both remains almost same. So if you're going to see, you can see 2 circles exactly at the center of the flagella or cilia. That's called as a central microtubules. And you can see a sheet which is coming out of them, which is an inner sheet. Okay. Outside you can see two circles combined. So they just called as a double microtubule. For example, A tubule or B tubule. And you can see a small hair-like things which is coming off the circles. They call as dyne arms. And the outermost membrane is always called as a plasma membrane. And the connectivity between the 2 round structures is called as a nexin. So this is a pretty much small diagram. And you can easily remember them if you're going to just remember a wheel of a cycle. Okay, right? In fact, if you're going to look at the diagram, the straight line which comes from the circles to the center, they call as a radial spokes. So this is pretty much easy to remember. The only thing is you just want to remember the labels and how they named. So I guess you would have liked this video. Thanks for watching this video. Please like, subscribe to our channels, and if you have any doubts, you are always welcome to put them on the comments. Share with your friends, and have a wonderful day.